Well, good afternoon, everyone. My name is Joel. It's really an honor to be back here in person at this meeting. I'd like to thank the CJD Foundation for all the wonderful work they do and for inviting me to speak today and sort of give you a complimentary lecture on some of the key concepts in prion disease. And I'll reiterate a few of the most important things that Brian already talked about, but I'll also talk to you about some of the more fundamental techniques and sort of areas of research that will directly be involved in the talks you'll hear tomorrow. So I'm also going to sort of put into context where each of those 10 talks sort of fit in the grand scheme of prions. And I also like to say that I wear two hats at this meeting. I'm up here presenting to you about prions, but I'm also a family member. I lost my father to sporadic CJD. He died in 1990 at the age of 39, so one of these sort of earlier onset sporadic cases. So first of all, I just want to sort of put prion disease in context and reiterate that they're a member of a family of related human diseases called neurodegenerative disorders that affect the brain, and these include much more common diseases like Alzheimer's disease and Parkinson's disease and ALS, also called Lou Gehrig's disease. And these diseases are more common than you may think. So they all feature loss of cells in the brain, and this is depending on which area of the brain is affected, that's why you get the different symptoms that typify each of these diseases. They're all progressive diseases, meaning that they get worse over time, and they're all age-related, meaning that they're much more common in older people. The common denominator of all these diseases is that they all feature protein misfolding and degradation. Now, where prion diseases are unique is that prion diseases are the only one of these disorders that is potentially infectious. So in my presentation today, I'm going to basically cover two topics. Go over again what is a prion, covering some of the same topics that Brian already talked about, and then talking about how we study prion disease in a laboratory environment. All right, so first, what exactly are prions? Well, let's talk briefly about prion disease, because Brian already talked about this. Because everybody is here at this meeting about CJD, we probably care more about the human prion diseases, but it's important not to forget that these are originally identified and studied widely in animals. And so the three most common prion diseases in animals are bovine spongiform encephalopathy, more commonly called mad cow disease; chronic wasting disease, which is a disease of cervids, which are your elk and your deer and your moose, and this is a disease that is spreading very widely in certain areas of the United States and Canada. And then the sort of most widely studied prion disease throughout history is scrapie, which is a disease of sheep, and it's called scrapie because the sheep lose their ability to have coordinated movement, and they end up having to prop themselves up against fence posts, and in doing so, they scrape off their wool. Now, the human prion diseases, you've heard about these. They're broken down into the three different classes: the sporadic, so that's sporadic CJD and VPSPR; the genetic or heritable versions, meaning that they can be passed on from generation to generation: fatal familial insomnia, GSS, and familial CJD; and then the infectious, or sometimes called acquired prion diseases, such as Kuru due to consumption of human tissue, variant CJD linked to mad cow disease, and iatrogenic CJD linked to errors in medicine. So I find prion diseases very interesting, not only because they have affected me personally, but they've attracted a lot of attention in the research community because they go against a lot of the norms that you see in biology. So first off, they're the only type of disease that can occur in these three different ways: sporadically, genetically, and via an infection. So things like viral and bacterial diseases, they occur via an infection. Something like cancer can occur sporadically or genetically, but it can't occur via an infection. As Dr. Appleby mentioned, for the infectious versions of the diseases, it takes an incredibly long time from exposure to the development of symptoms, sometimes on the order of decades, whereas with a viral or a bacterial infection, you're talking a few days to a few weeks between exposure and symptom onset. Now, while all prion diseases are potentially infectious, very few so are naturally infectious. So none of the human prion diseases are contagious per se. But there are prion diseases that are naturally infectious, so they can spread horizontally from one animal to another, and that's CWD and scrapie, and we still don't know exactly why and how these diseases can spread from animal to animal. And lastly, prions are unlike viruses and bacteria in that they're very hard to destroy. So a lot of the common techniques that you would use to sterilize prions in sort of a medical environment, or sterilize bacteria and viruses in a medical environment, don't work so well with prions. And this is why sometimes you get these errors in medicine that lead to iatrogenic transmission of prion disease. So just a quick review of biology. So this is kind of like high school biology or first year university. We have DNA in our cells. These are our genes. DNA gets transcribed into RNA, ribonucleic acid, and then ribonucleic acid gets translated into proteins. And proteins are the workhorses of our cells. They do things like catalyze the enzymatic reactions necessary for metabolism. They form the structural building blocks of the things in our cells that give them their shape. They regulate things that get inside and outside of our cells, so they're basically the main sort of the doers in the cell. So here's the gene sequence for the prion protein. The gene's called PRNP. There's a test at the end, so start memorizing. So this gets translated into prion protein. There's the prion protein gene sequence. So proteins are made up of building blocks called amino acids. There are 20 different amino acids. They first start off as like a beads on a string, and then once the full string is produced, then they fold up and adopt their three-dimensional structure or fold, and that fold or structure or shape is what gives them their function. So tomorrow, one of the talks is going to be by Dr. Shashi Shetty, who is going to talk about how variations in the amount of DNA, either gain or loss of DNA in certain regions of the brain, might be a risk factor for developing certain types of CJD. So what exactly is a prion? As Brian mentioned, prion comes from the words proteinaceous infectious particle. If you put those together, you get proin. That doesn't really have a nice ring to it, so you rearrange the I and the O to get prion. So sort of the main thing about prion disease is you have a protein that can exist as two different shapes. You have the PRPC, or cellular shape. That's the good shape that is healthy, benign. We all have it in our brains. And then there's the disease-causing or scrapie isoform PRPSC, which is the infectious and toxic version. And prion diseases, they buck a lot of trends, and people had a hard time believing that these were the cause of diseases like CJD and scrapie, that it must be caused by some sort of weird atypical virus. And that's because they go against two sort of fundamental concepts in biology. The first is that if you have a protein, it can only have one shape. Well, here's a protein that can have two shapes. Secondly, the idea that a protein could be infectious was just heretical. And there are still some people out there today who are convinced that prion disease are caused by a virus and not these misfolded proteins. Because these diseases are so interesting, a lot of people have started researching them, and there have been 2 Nobel Prizes awarded for prion disease. Carlton Gajdusek first showed that Kuru and CJD are actually transmissible or infectious diseases. And then probably most well-known, Stan Prusiner won the Nobel Prize for sort of coining the term prion and outlining this infectious protein hypothesis. And I'd like to say that there's a third sort of honorable mention prion prize too, as Kurt Wuthrich won it for a technique called NMR spectroscopy, which is used to determine the shape of a protein. But the most famous protein shape that he determined was the prion protein. Okay, so what exactly is a prion and how does it work? And some of you who have watched my talk I recorded for the foundation last year may have seen this slide already, so don't spoil it for others. So if you think of PRPC as a car, a Chevrolet Corvette, so like other proteins, PRPC is a dynamic entity. You can undergo slight fluctuations in its structure. So for instance, it can open its roof, it can open its doors, it can open its hood, but it's still a car. You can still tell it's a car. So what you have happen in prion disease is you have conversion to a structure that is fundamentally different and one that is dangerous to a cell. And so this transformer analogy, I think, is appropriate, but from a slightly more scientific standpoint, here's what's going on. So you have PRPC on the left. Those red regions are called alpha helices. They're sort of a corkscrew shape. They get converted into a protein that is rich in the blue element there, and those are called beta sheets. And then once you have this, go back one, yeah. Once you have, there we go, this being produced, this does not exist on its own. It immediately clumps together to form these large aggregates that then deposit in the brain. And the reason why prion diseases are potentially infectious is because if you take PRPSC and it meets a molecule of PRPC, it causes PRPC to adopt the misfolded version. It becomes, as Dr. Appleby said, the bad kid. And this property is called self-propagation or self-multiplication. And then one of the talks tomorrow from Leo Cortez, he's going to be talking about how there's actually a spectrum of sort of sizes of clumps between this, and how those different sizes of clumps may be responsible for some of the variability that you see in prion disease. So there are these three types of prion disease, and I'm not going to spend a lot of time on this, but how they are thought to occur. The sporadic is that you have sort of a rare spontaneous misfolding event in the brain where PRPC gets converted into PRPSC. And in all likelihood, you probably have to have several of these events occurring at the same time and in the same place in the brain for you to form that sort of seed of PRPSC that can then multiply. And this is why prion diseases are so rare, because this event is thought to be vanishingly rare. Oops. The second is in the genetic prion diseases, you have a mutation in a gene, and that causes you to produce a slightly different prion protein. And this change in the sequence of the prion protein is thought to promote the misfolding of the prion protein, making it more likely that it will adopt the bad form. And then lastly, in the acquired or infectious forms, you have an introduction into the body of an exogenous source of preformed prions that can then convert the host prion protein into the bad form. So in the genetic prion diseases, there are many different mutations that are known to cause genetic prion disease. Some of the most common ones are E200K, which causes CJD, D178N, which can cause a form of CJD, but more commonly causes fatal familial insomnia, and then P102L, which is a cause of GSS. Just to give you one example of how this works, for fatal familial insomnia, the normal DNA at position 178 is GAC. In FFI, it gets mutated where the first G becomes an A, and whereas GAC instructs the cell to put a aspartic acid, or D, in that position, AAC causes the cell to put an asparagine, or N residue, and that difference in the sequence then makes the protein more prone to adopting the bad form. So this is the prion protein. As Brian mentioned, the shape of a prion or a protein determines what its function is, and we're not really sure what the PRPC does in the brain. What we do know is that it's found on the outside of a cell. So here's a cell. The surrounding part of a cell is called the lipid bilayer or cell membrane. So the prion protein is attached to the outside of this, so it sort of faces the outside of the cell. And one of the presentations you're going to hear from Dr. Altmatman is about what happens when you cleave off the link between the protein and the cell, causing it to float away, and how that might be something that can be exploited for therapeutic purposes. So as I said, we don't really know what PRPC does. It can't have evolved solely so that 1 to 2 new cases per million people could get prion disease every year. So it has to do something useful in the brain because it's present from fish and birds and frogs, and all mammals have it. So it's still one of the mysteries as to why we have this protein. In all likelihood, it probably plays sort of more subtle roles in several different areas of biology, as opposed to a major role in one area. All right, so moving on to the second part of my talk, and that is how do we study these diseases in the lab? And probably the most common way is by what's called using the mouse bioassay. So to study prions, we take advantage of the fact that they are infectious. And so you take a normal old mouse and you inject it with a known source of prions. And most commonly, you inject it directly into the brain because this is the most efficient way to initiate the disease. But in reality, you can actually put it basically anywhere in the mouse's body, and it'll eventually make their way up to the brain. So then you wait. This is one of those survival curves that Dr. Appleby talked about. So after a period of about 4 to 5 months, the mice will start to get sick. They'll show signs of neurological impairment, they'll lose weight, their backs will become hunched, their movement will become uncoordinated, and eventually they would die of prion disease. And then if you look at extract from their brain material, and you use an antibody that specifically detects the prion protein, and you're going to see lots of things that look like this weekend. So in healthy mice that don't have prion disease, this right here is PRPC, the normal prion protein. If you treat this with a different protein called proteinase K, so this is a protein whose job it is to chew up other proteins and convert them into tiny little pieces that can then be recycled to make other proteins. So because PRPC is not clumped together, this enzyme called proteinase K, or PK, gets rid of all of it and it goes away in healthy animals. In prion-infected mice, because the prion protein is in its scrapey form, PRPSC, these are clumped together to form these large aggregates. The protease can't chew them up, they're just too big. And so you get resistance, and so the signal does not go away after digestion, and you're left with things that look like this. And these 3 dark regions here, these are the biochemical signature of prion disease, and you'll see this a lot. If you look at the brain tissue itself, you'll see the pathological sort of hallmarks of prion disease. These holes in the brain is called spongiform degeneration to give it a Swiss cheese-like appearance. And all this brown stuff is the PRPSC that's formed these deposits that are interfering with the cell function. And prion disease, although we're all very unlucky to be, had a loved one affected by these diseases, the animals systems used to study this are better than for any of the other neurodegenerative diseases. So for Alzheimer's disease, Parkinson's disease, these are mice that have prion disease. They're not even a model. These are mice with prion disease. So we have a very good way of studying these disorders. So the prion protein, you have to have it or else you can't get prion disease. So if you take mice that have been genetically engineered so they no longer produce PRPC, you inject them with prions, they're fine. They live their normal 2-year lifespan. So you have to have PRPC. And this is one of the good sort of therapeutic strategies for combating this disease, is getting rid of the substrate. If you have no substrate, there's nothing to replicate, and you get no propagation of prions. So this system that I just talked about, you inject mice with pre-existing source of prions. This is the most common way of studying prions in a laboratory environment. The problem is, in humans, 99% of cases do not arise due to an infection. They arise either sporadically or due to a mutation. So there's no pre-existing source of prions that gets introduced in the body. And studying this in mice has proven to be much more challenging, and it's something that's still being worked on. So one of the talks tomorrow is from Dr. Joaquin Castilla, who's going to be talking. He's an expert in actually both of these different types of mouse models. And he's going to be talking about his really exciting work on developing a paradigm for studying GSS using mouse models. All right, so prions are potentially infectious, but they're not uniformly infectious. So for instance, if you take mice and you can try to infect them with prions that aren't from mice, so human prions like CJD or chronic wasting disease prions from deer, it doesn't work. And so you inject these mice, they will live probably 2 years and maybe only 1 or 2 will get sick, and it takes way longer than normal. And this is because what's called the species barrier. Basically, the idea is that prions from species A do not efficiently infect animals from species B. This is a good thing, because in our lab, it allows us to study, say, mouse prions without worrying that we're going to accidentally inject ourselves and give ourselves prion disease. Now, in reality, there's a spectrum of prion disease susceptibility. So things like mice and hamsters are kind of in the middle. On the very resistant side, you have animals like rabbits and horses and dogs, which are almost or completely resistant to prion disease. At the other end, you have the bankful, which, for those of you who've seen me talk before, I'm the bankful guy. Dr. Castilla is also a bankful guy. And these are animals that seemingly don't have a species barrier. You can infect them with prions from anything. So they're a very useful tool for prion disease research. And why do we have a species barrier? Well, it's because of differences in the sequence of the prion protein gene and protein between different animals. So there are slight differences in those arrangement of amino acids between, say, the human prion protein and the mouse prion protein. To illustrate how this works, if you take CJD prions, inject them into mice, which produce the mouse prion protein, mouse PRPC, you don't get transmission because of the species barrier. If you genetically engineer the mouse so that it now produces, instead of the mouse prion protein, it produces the human version, then the mice become very susceptible to CJD prions and you get efficient transmission. So to have good transmission, you have to have a match between the PRPC in the system you're trying to replicate the prions in and the PRPSC that you're using as your test sample. So not all prions are alike. There are different strains of prions, much the same way that there are different strains of viruses or bacteria. Instead of having different or changes at the nucleic acid level in prion disease, the changes at the structure or shape level. And so it turns out that for a given PRPC that it can actually misfold into several different shapes. And each of these different shapes then cluster together to form aggregates, and they give slightly different phenotypes or different they have different properties in animal models, and you can differentiate these based on the symptoms they cause, how long it takes them to cause disease, differences in biochemically after treating with enzymes like proteinase K, and different neuropathologies. And so these are related to different shapes of PRPSC, and this has now been proven using this really cool technique called cryo-electron microscopy. So studying prion disease in mice is great. The drawbacks is that, of course, we're using animals, and we'd always like to reduce the number of animals we use in research. It also takes a long time and costs a lot of money. And so we're always looking towards simpler systems that we can use to study prions. And one of the main ones are cells. And so you can culture certain types of cells in a petri dish, expose them to prions, and then they'll become infected with prions. Here's that biological signature of prions again. And they'll maintain that infectious status indefinitely. And you can use this to do things like study how PRPSC is made, how protein misfolding works. More importantly, you can use it to identify drugs that might interfere with this process and could therefore be therapeutics for CJD. Now, that's sort of in the first generation models. Now with sort of advances in genetic engineering, how many of you in the audience have heard about something called CRISPR in the news? Yeah. So CRISPR is a new gene editing strategy. So what you can do is you can take a cell line like these CAD5 cells that they're a mouse cell line, so they produce the mouse prion protein, and therefore they're only susceptible to mouse prions. So if you get rid of PRP in these cells by using this CRISPR gene editing approach, and then you can put back in whatever prion you want. So you can put back in, if you put back in mouse PRP, the cells are susceptible to mouse prions. If you put back in the hamster prion protein, they're susceptible to hamster prions. Or if you put back in the bankful prion protein, which has no species barrier, they're susceptible to both mouse and hamster prions. So things like this are going to really sort of, I think, accelerate our ability to discover small molecules that counteract prion replication. And even sort of more cutting edge, and you're going to hear a talk about this tomorrow, are these so-called cerebral organoids. So you can take stem cells and you can convert them into brain cells, which then sort of aggregate together and form what are called mini brains. So it's like a miniature human brain that you can study in a petri dish. And the nice thing about this is that these are human sourced tissues, and so they sort of more or better reflect the human situation. And so Catherine Haig's lab has shown that you can infect these with CJD prions, and tomorrow you will hear a talk from Bradley Groveman, who works with Catherine Haig on how these cerebral organoids are really advancing our understanding of human prion disease. Okay, so getting near the end now, I'll mention this quickly because Brian already talked about RT-QuIC, but the sort of two main ways of studying prion disease in a test tube are called RT-QuIC and PMCA. And the reason this is beneficial is because animal experiments take months to years, cell experiments take weeks to months. This only takes hours to days, so this is much faster. The idea is the same thing. You have PRPC and PRPSC mixed together, prion replication occurs, and then you use something to break up your growing clumps into smaller pieces. And the reason you need to do that is you think if you have, let's say you're building a tower out of Lego bricks, and you have two choices if you want to make it bigger. You can add one to the bottom, or you can add one to the top. However, if you break that rod into 3 pieces, now you can add 6. You can add one to the top and bottom of each of the 3 pieces. And so in these assays, you have something that breaks apart the growing aggregates into smaller chunks to allow for exponential replication or amplification, which allows these things to be done in hours or days. And so the difference between PMCA and RT-Quick is the way that it gets done. So in PMCA, the aggregates are broken up using sound energy or sonication, and with RT-Quick, they're broken up using vigorous shaking. And these techniques are incredibly sensible. They can detect prions in, as Brian said, in CSF, but also skin, urine. And there are going to be several talks on this. One from Marcelo Varilla, who's going to talk about using PMCA to detect PRP-SE prions in appendix samples. And then one from Jamie Noguez, looking at cases of RT-Quick that are neither positive nor negative, but somewhere in between, and learning how to deal with those. Okay, so there are still no therapeutic for prion disease, but we're getting closer every day. So if you break prion disease down into sort of its simple chemical equation where you have PRPC turning into PRP-SC that then causes disease, there are several different opportunities for intervention. As I mentioned, you could reduce PRPC expression using something like antisense oligonucleotides, an approach that's championed by Sonia Valov and Eric Minikal, who are in the audience today. Or you could do some other things. So you're going to hear a talk from Rodrigo Morales, who is using PMCA to identify small molecules that inhibit the conversion of PRPC to PRP-SC. You're going to hear a talk from Zurui Wang that talks about these really interesting compounds called cellular ethers that we don't know exactly how they work, probably by breaking up pre-existing aggregates so that they can be cleared. And also Maria Letizia Barreca, who's going to talk about identifying compounds that block the neurotoxicity associated with prions. So just to summarize some of the key concepts that I've gone over. So prions are misfolded and infectious protein aggregates that cause neurodegenerative diseases. They're solely composed of PRP-SC, which is a misfolded version of PRPC. PRP-SC can self-propagate or self-multiply, which is why prion diseases are infectious. A species barrier means that you have to have a good match between PRPC and PRP-SC. And prions are best studied by taking advantage of their infectious properties, either in an animal model, a cellular model, or by using in vitro techniques such as PMCA or RT-Quick. Despite all these things, there's still a lot of unsolved mysteries in prion disease. We don't know why, for instance, prions kill brain cells. That's still one of the biggest mysteries. We don't know what PRPC does. And I guess perhaps most importantly, we still don't have a therapeutic that works in humans. I just want to leave you with sort of an interesting point. And so prion diseases are rare, certainly they're more rare than Alzheimer's disease and Parkinson's disease. However, some of the fundamental game-changing things that have affected the biology of Alzheimer's and Parkinson's recently have originated in fundamental research in prion disease. So just to give you one example, RT-Quick, developed by Byron Coe to amplify and diagnose prions. This has been modified and adapted for use in Alzheimer's disease and Parkinson's disease, so that for Parkinson's disease, it is now possible to diagnose Parkinson's disease in patients using blood samples. And this is in living patients, so ante-mortem. Thus, fundamental prion disease research can lead to major advances into much more common neurodegenerative disorders, which is why a message that you should be telling your representatives is that funding prion disease research and studying prions is and will continue to be important. So with that, I'm going to stop and we'll take any questions along with Brian, and then there's going to be a